First of all, thanks for the organizers for the invitation yesterday, and then this talk. I was talking about yesterday about our approach to use a new type of endothelial cells for vascularizing cardiac tissue, and using these cells as a supplementary cell type to support cardiac transplantation of various constructs you can make from pluripotent stem cells. So what I showed yesterday, and just briefly touch upon that, is that we can make very high fidelity endothelial cells, manage to scale up the production of cells. And the importance of having these cells in hand is that we can have a second cell type which can support vascularization and, first of all, feeding these constructs, and would overcome a major hurdle we facing for the last decade or so, I would say, is that whenever we try to implant cells, cardiomyocytes, other cell types into the injured heart, those cells don't survive. Those cells are lost within days or weeks. And we had a feeling if we support these cells with another cell type, like these endothelial cells, that these constructs would be more successful and would be more easier to translate these into clinical applications. So this was the background of my presentation, and the few models I presented there was models to show that these cells can form vessels. In small animal models, these cells can be used for making vascular grafts, which can be implanted by surgeons to replace injured vessel areas. And I also showed some cardiac examples when we're mixing endothelial cells with cardiomyocytes and make those into a cardiac constructs, 3D construct like engineered heart tissues or other cardiac grafts, that these can be implanted successfully on top of the heart, with the hope that the heart failure issues could be solved, and at least in a preclinical setting. And our hope, again, as mentioned, that we can scale up these productions and we can have more information about these cells, so we can take these cells into first-in-man studies.